Thank you, Conway, for the introduction, and thank you everyone for joining. So today I will be presenting our new work on defining the spatiotemporal and the molecular trajectories of beige adipogenesis. Okay, so our bodies have a remarkable capacity to adapt to external cues. For example, in response to temperature fluctuations, in response to nutrition, during feeding, fasting, or during exercise. However, changes in lifestyle, where more sedentary behaviors are introduced, or overnutrition may lead to unhealthy adaptation. This unhealthy adaptation will promote health complications such as obesity, type 2 diabetes, and cardiovascular diseases and fatty liver. In the lab, we're interested on understanding those healthy adaptation processes and unhealthy adaptation processes through the lens of adipose tissue remodeling, with this idea that if we are able to identify pathways that are changing during the adipose tissue remodeling, healthy or unhealthy, we can use those and leverage them to reprogram the tissue and restore metabolic health. So adipocytes are three different subtypes. White adipocytes, which are those unilocular cells, they have low content of mitochondria, and their main job will be to store energy and release it in times of need. Brown adipocytes are the second type of adipocytes, and those are multilocular cells. They appear under the microscope as multilocular cells, and they have a high content of mitochondria. This allows them to be more thermogenic, and this means that those cells, in response to cold, for example, they'll be able to produce heat. They're mainly developed during embryogenesis, and they're located in infants in this intrascapular region or perirenal region. There was believed for a long time that those adipocytes actually disappear in adults. The third and last part of adipocytes are those beige adipocytes or bright adipocytes. They are characterized, they share the characteristic of both brown and beige fats. And they are as well thermogenic, they can produce heat, and they are high in mitochondria. And most importantly, and maybe this is the key part, they are inducible, they're highly inducible. So they can be induced, for example, during cold stimulation or adrenergic stimulation to contribute to maintain the heat, to produce some heat and maintain the body temperature. So as I said, brown fat was believed for a long time to have disappeared in adults. However, recent works from different groups using PET-CT scans. So this is basically imaging approaches that can allow to detect metabolically active regions that are able to uptake glucose, have shown that, for example, here we can see one PET-CT scan in thermal neutrality. You can see that the patient do not have any active region that uptake glucose. However, when the groups exposed people to cold, they saw that there was an increase of uptake of glucose in different regions surrounding the neck and the shoulder, suggesting that there was some brown fat present in those people. More interestingly, they found that in obesity, brown fats completely disappeared. And this is even when the people were exposed to cold, suggesting that maybe a link between brown fat and obesity. And maybe brown fat would be protective against obesity. And this was indeed the case. So recently, a group from Paul Cohen group went back and did a study, a retrospective study review of all PET scans that has been done in a hospital, and they used about 50,000 patients, and they classified basically those patients depending of they express brown fat or not. And they looked 10 years down the road, what was the metabolic outcome for those patients? And actually they found that patients were protected against type 2 diabetes, dyslipidemia, and cardiovascular diseases, suggesting that brown fat was actually protective against cardiometabolic diseases. Moreover, more recently, Cyprus group used a drug that was approved by the FDA that targets specifically beta-adrenergic receptor. And they treated patients, so here patients that do not express brown fat, and they treated them with this drug for 28 days, and they performed PET scans before and after. So before there was no expression of brown fats or no glucose uptake, and you can see the treatment with 28 days were able to restore this brown fat either by recruiting new beige fat or by inducing the activation of this brown fat. This overall suggests that thermogenic fat is important for metabolic health, and that brown fat could be induced actually in humans, so making it very relevant for human health. So the main question that we were interested on was to understand how this brown fat appear, and more specifically is to understand those early stages that control and initiate this brown or beige biogenesis. To do so, so as you know, adipose tissue is a very complex organ. It's actually composed with different cell type. White adipocytes are the major cell type within the tissue. But actually those represent, in number, they represent only 30% of the tissue. And the other part that we call stromal vascular part, or the stromal cells, are is composed by pre-adipocytes, mural cells, immune cells, and endothelial cells are the majority of the tissue. So to be able to dig in more in details on how beige adipogenesis happened, we decided to use single cell sequencing. So we designed an experiment as follows. So we took mice and we either maintained them at room temperature or treated them with beta-3 agonists or with cold. And we chose a window of 3 days treatment with this idea to look clearly at this early stages of progenitor cells changing. We isolated stromal vascular fraction, and then we depleted from all the immune cells. Then we submitted to 10x genomics. So to our surprise, our first finding when we compared between different treatments, we didn't find any differences between the subpopulation that are present in the three conditions. So whatever treatment we use, and they were looking the same. So the same population of cells were present compared to vehicle treated mice. And even though we can see that cold and beta-3 agonist treatment can lead to browning of the tissue. However, what we did notice is that there was actually changes in the cell numbers of each population. And more interestingly, we found that mural cells are more induced. So the number of mural cells were increased in cold condition compared to beta-3 adrenergic stimulation, where mesenchymal cells and interstitial cells were down-regulated. The numbers was decreased in cold condition compared to beta-3 adrenergic, suggesting that there will be maybe some differences between those two treatments that lead to the same phenotype, which is the browning of the white fat and the recruitment of those beige adipocytes. So to dig in more details on this cellular dynamic that's happening within the tissue, we decided to perform a pseudo-time analysis. So this approach is basically takes advantage from the fact that the cells within the tissue are a continuum of states, and those cells could be bioinformatically reordered to predict the fate of the cells or the destiny of cells through time. So we did find that one single population that is here was able actually to give rise to multiple cell lineages. Now this population was mural cells. This is consistent with previous reports suggesting that mural cells can give rise to multiple subpopulations. So to confirm this finding, we next performed some gene ontology analysis. And we found that this population of cells was indeed enriched from pathways that are associated with mesenchymal cells development and cell phase determination, suggesting that our analysis and finding that those mural cells give rise to those different pathways is right. So we next wanted to zoom in and look in more details to what's happening within each lineage. And so we compared between the density of different cells that will contribute to each phase. And then again here, we found that depending on the treatment that we are looking at, there was an enrichment that changes between each treatment. So and the contribution of each lineage, each cells, or the numbers of cells clusters to each lineage was changing. And this was true as well when we compare between beta-3 adrenergic stimulation and cold. So we next reordered the differentially expressed genes depending according to their hierarchical clustering on cold conditions. So we took basically just the genes that are expressed in cold condition and reordered them. This is an example for example, for lineage 2. And then we forced that ordering, the same type of ordering in the other conditions. So in beta-adrenergic stimulation and vehicle. What we found is actually there was differences again here in the pathways that are driving the movement within that lineage 2 in cold condition compared to other conditions. So we wanted to understand what are those major pathways that are driving this beige adipogenesis. So to do so, we did some pathway analysis, and then we found that some major pathways that drive this beige adipogenesis in cold conditions are associated with fatty acid transport, actin cytoskeleton rearrangement, and extracellular matrix organization. This is consistent with previous reports. One of the pathways that we were not expecting was the response to enfolders protein. So to remind you, this pathway involved the activation of 3 different Rs, and it's regulated with this protein that's called BIB that's expressed within the ER. And basically, when there is an accumulation of misfolded proteins, BIB will recognize those misfolded proteins and then will release the activation of those 3 different Rs. So ERE1, for instance, will activate XBP1, and PERK will phosphorylate ERF2, that itself will activate ATF4, and then ATF6 will translocate to the Golgi and be activated there. As a readout of the activation of the enfolded protein response, we used a phosphorylation of ELF2. Then we stained mice, tissue chemical mice that were treated either with cold or with the beta-3 agonist. And then we can see that that under cold only, there was a phosphorylation of ELF2, where we didn't see it in beta-3 adrenergic agonist activation or vehicle, suggesting that actually that there was some differences between the pathways that are driving the browning process in beta-3 agonist and cold. So to summarize this first part, using bioinformatic approaches, we were able to identify that there was no changes in the cell population within the early stages of beige adipogenesis. We also found that there is dynamic changes within the cell numbers, suggesting that specific phases were promoted depending on the treatment that we are using. And finally, we found that there is no differences between the cellular progression of mural cells in response to cold exposure and beta-3 adrenergic stimulation. So to dig in and understand more and validate those findings, we went back in vivo. And we decided to answer the first question, which was, are those changes within the cells maintained in vitro? And to do so, we basically took mice, treated them with beta-3 adrenergic or cold or vehicle for just 24 hours. And then we isolated those progenitor cells. We performed gene expression for genes that before differentiation, and then we took those cells and differentiate them with a standard cocktail that will give rise to classic adipocytes. And then we performed again the same gene expression. And we found that for undifferentiated cells, we found consistent with our finding that there was actually an induction of brown markers, HCP1 and DAV2. We also found that those cells are actually maintaining this phenotype when they are differentiated in vitro. suggesting that the cells are actually primed to this beige phenotype in vivo. And that this priming process is actually maintained even when the cells are removed from the tissue. So next question was, can we actually follow those cells and in vivo and predict that this is what's happening really in vivo? So we went back to our single cell sequencing, and then we looked to markers, gene markers that are expressed within those mural cells. And we found that SM22 transgene is highly expressed in those cells. So we generated a mouse that enable us to do lineage tracing in vivo. And basically those within the tissue, when the mice are fed the doxycycline, it start to express GFP in mural cells. This will allow us to follow what have destiny of those mice if they are treated with different treatment. So we took the mice and we treated them either with beta-3 adrenergic or with cold. And then our first question was, are those cells, is those treatment inducing any expansion of the tissue or proliferation of the cells? So we added EDU, which is a marker that incorporates and reflects the proliferation of the cells in vivo. And we performed some flow cytometry, and you can see that under treatment of beta-3 adrenergic agonist and cold, there was an increased proliferation of progenitor cells or SM22 marked cells. However, the increase was surprisingly less important in their cold condition when we compared with beta-3 agonist stimulation. So we scratched our head for a little bit before finding out that actually when we look to the total number of progenitor cells within the tissue, it didn't change. So we had a change in the progenitor cells number that are marked to GFP, but few population of cells had been marked to GFP, where the other cells that were not marked are still proliferating, suggesting to us that actually those cells, we are not able to find them anymore because they are recruited actually within the tissue. So to validate this, we took tissues and then we stained for GFP after 3 days of exposure to cold or beta-3 agonist. And we can see that indeed the treatment with cold do induce the recruitment of beige adipocytes, where beta-3 agonists didn't induce the recruitment of SM22 positive adipocytes or progenitor cells. So to summarize this second part, we found that under cold condition, SM22 positive mural cells are recruited, are first primed to this beige phenotype, and then they are recruited to those beige adipocytes. However, when we treat with beta-3 agonist stimulation, we actually see that those cells can still prime. So the same processes of priming are probably happening in this condition, but they're accumulating and not recruited to the beige phase. So, so we wanted next to know how this is happening, why those cells are, what is the mechanism that allows the cells to be recruited that's missing in this condition of beta-3 agonist. So to determine what these pathways are, we first wanted to identify what were the conditions, the minimum condition that will lead to less proliferation of the cells. With this reasoning that if we want really to identify the pathways that are leading the recruitment, we need to identify the early stages of this stimulation. So our readout was cell proliferation. So we treated mice with beta-3 agonists at different times for either 1 day, 3 days, or 7 days. And then we injected them with EDU that will reflect the proliferation of the cells. And then we can see that actually at 1 day, there was no proliferation. And this proliferation started to appear at 3 days and was further enhanced at 7 days. So we decided to analyze gene expression at 1 day. And we sorted the cells and then we did an RNA-seq. And you can see that treatment with 3 days induced a certain number of genes that were induced. One of those genes that was very interesting to us was induction of beta-1, ADRB1 gene. So we confirmed this, its expression we treated mice with beta-3 agonist or cold, and then we isolated RNA. You can see that there's indeed an increased expression of beta-1 receptor. So to implicate beta-1 in this pathway in the recruitment of the SM22 positive cells, we decided to go back in vivo, and then we resonated that if this pathway was important in the recruitment of those SM22 positive cells, it should be able to inhibit the recruitment of beige adipocytes when the mice are exposed to cold. So we took mice, we exposed them to cold, and we treated them with beta-1 antagonists to block the pathway. And indeed, this treatment led to a complete abolition of the recruitment of beige adipocytes from SM22 cells in 4 degrees. So we next wanted to do the opposite. So we took mice and then we put them at terminal neutrality, which means that we are blocking completely any catecholamine secretion and adrenergic input. And then we treated the mice with beta-1 agonist. So the expectation here that if we combine beta-1 agonist and beta-3, we should be able to recapitulate the cold conditions and the recruitment of the SM22 positive cells. And indeed, this was the case. So when we treated the mice with both at terminal neutrality, we were able to induce the recruitment of the SM22 positive cells, and this is within 3 days of treatment. So to summarize this third part, we can see that we show that the treatment beta-3 agonists induce the priming of progenitor cells. The cells, as they are progressing through the beige phenotype, they start expressing beta-1 receptor. The expression of beta-1 receptor will be important for the recruitment of cells. So because it's not present in this just beta-3 agonist stimulation, we don't see recruitment of those cells, where under cold conditions, the presence of catecholamines that are secreted through the neurons is able to induce the recruitment to the maturation of those progenitor cells through beige adipocytes. So our next question was, okay, so those cells are able to be recruited. However, what is the first inducer? How those cells are primed to this phenotype? So we went back again to beta-3 agonists, and then we know that beta-3 receptor is only expressed in mature adipocyte and it's not present within the stromal vascular fraction. So we decided to take mature adipocytes after treatment with beta-3 agonists and then isolated cells and performed an RNA-seq in those cells. And we see that there was an increase and then an increase for a number of genes. We analyzed we did some gene ontology, and we see that, as expected, major pathways that are induced are associated with fatty acid metabolism. But we noticed that there was an increase of those omega-3, omega-6 metabolism, alpha-linolenic pathway metabolism. So just to remind you, those pathways are the breakdown of linoleic acid and alpha-linoleic acid, and they can give rise to a variety of intermediary. The last ones are arachidonic acid for linoleic acid, and EPA and DHA for alpha-linoleic acid. And those product free fatty acid product can be released in the microenvironment. So we decided to treat cells that are isolated from mice with those compounds, with those lipids, and then see if we are able to induce changes in progenitor cells phase and inducing beige adipogenesis. So the pre-treatment with arachidonic acid and DHA actually was able to induce browning. We didn't see any effect for EPA, but this suggested to us that free fatty acids that are released in response to beta adrenergic stimulations are able to prime those beige, the progenitor cells, SM22 positive cells, to the beige lineage. So to confirm that in vivo that lipolysis is implicated in these pathways, we decided to take mice and put them in terminal neutrality and block lipolysis with this compound called acipimox, and treat the mice with beta-1 and beta-3 agonists. Now that we know that both of them are necessary for the recruitment of beige adipocytes. So we found that indeed the inhibition of lipolysis completely abolished the browning, the recruitment of beige adipocytes, suggesting that lipolysis, free fatty acids or the products of lipolysis are necessary for this recruitment of SM22 positive cells. So to summarize, we found that during cold exposure, the activated secretion of catecholamines will act in mature white adipocytes through beta-3 agonists. This will lead to the secretion of free fatty acids. Those free fatty acids will act on progenitor cells, SM22 positive cells. This will now involve and move through the adipogenic lineage, starts expressing beta-1 agonists. The activation of beta-1 agonist is necessary for this, for the progression to beige adipocytes and the induction of browning. So in conclusion, the combination of both single cell and in vivo experiment, we were able to define the different discrete steps that can lead to beige adipogenesis. Finally, I want to thank the people that contributed to this work. Steve Farmer for his support, and the funding, different funding agencies, and the dkNET for their support. And I'm ready to take any questions.
Okay, thank you for Nabil. Now is our Q&A session. So if you have any question, please feel free, just use the chat or just unmute yourself. Yeah. Okay. I see there's a question from the chat. So great presentation. How are those SM22 mural cells related to previous identified PDG FRB mural cells?
Okay, so those so SM22 positive cells are do express PDGFR beta, but they're actually a subpopulation of the PDGFR beta positive cells. So they are more, PDGFR beta will mark both mural cells and mesenchymal cells, where those cells will mark specifically just mural cells that are expressed within the tissue.
Thank you. Any other questions from the audience? Okay. There is one question. Maybe I missed this, but how cold were the mice in the starting slides?
You mean when you treat them with cold? It's so we put the mice. Yeah, we put the mice at 4 degrees. So at the beginning they are at 23 degrees, so at room temperature basically, and then we move them at 4 degrees. Does this answer the question?
I think so. And
so there was recently identified populations from the Patrick Seeley group and Kristen Wolfram's group, a couple of subpopulations of progenitors like CD142 or DPP4, like, you know, some anti-adipogenic and some pro-adipogenic populations. Did you so how do these compare to the mural cell populations? And did you check, are those, you know, these SM22 positive, is it like a larger umbrella of these populations, or is it a smaller one? And did you check to see how in your conditions these identified populations change?
Oh, so yeah, so the DPP4, we see them, they're present there, and they're mesenchymal or interstitial cells. So I didn't have time to go in more details, but we do see them. The inhibitory cells, so the IREC cells, it's more discrete, but we do see some cells that do express some markers. the SM22, regarding the relationship, the SM22 cells are actually earlier than those cells. So they basically can give rise to at least the DPP4 positive cells.
I see. Okay, thank you. Yeah.
Yeah, and there is another question from the chat. Would you recommend at this point to experience levels of cold that could help drive down fat development?
I mean, I don't recommend to people to stay in cold, but if you can do it, well, why not? But clearly, cold exposure prevents at least most of the cardiometabolic diseases. The idea is now to be able to identify those pathways to leverage in humans and then use it as a treatment. I think some people do take a cold shower. I don't know if it helps, but.
Okay, the next question, how does the experiment translate to humans? Is it stress in general that can cause lipid metabolism changes?
So lipid metabolism changes. So for the browning, for the lipid metabolism changes, yes, the stress obviously will affect in different ways. It's not only through adipocytes, it's also through secretion of different stress hormones that will affect lipid metabolism. Now, whether it will induce browning, you need to be exposed to cold to induce browning. I don't know if I'm answering the question, but what do you mean by stress?
I think maybe it means addressed in the chat. Yeah. So Salon, is that answer your question? Okay, so Salon means cold is used as stress, right?
Yeah. Yes. So cold, you can see it as a stress. Okay.
Does that answer your question? Yes. Okay, I'm moving to next question. So it's from Kwam. The EPA and the AHA stimulate the cell. Beige was done in primary SVC differentiation to brown adipocyte. I think that's a question.
Yeah, so the experimental setup was we isolated cells from in vivo and then progenitor cells, and then we treated them with the DHA and AHA for 2 days, and then we differentiated the cells. And we assessed the expression of thermogenic markers. And we saw that both DHA and AHA were able to, the cells that were exposed to DHA and AHA were able to express more thermogenic markers, where the cells that were treated, for instance, with EPA were not able to express those markers.
Okay, I think. Okay, yeah, Juan said you answered the question. So any more question or discussion at this point? We have some time left.
I mean, maybe I can ask something else. In the single cell experiments you showed, so those were single cell, right? Not single nucleus.
Yes, they were single cell. Yes.
So are you capturing any mature adipocytes, and or is it only progenitors? Like, do you have any adipocytes that they look like beige or they look like white adipocytes?
No, we didn't have any adipocytes. So we did deplete completely from immune cells and enriched with ScRNA. So we don't see any adipocytes in this direction.
So you only took the stromal vascular fraction to see. Okay, all right, thank you.
Yeah.
Okay. Any other more questions?
Maybe can I ask another last question?
Sure.
Sorry. So there was also from the Yuhua Cheng lab at Joslin, they identified these TRPV1 positive cells, and also via trajectory analysis, they could show that they lead to some beige cells. Could you see, like, how are they related, and could you see the TRPV1 positive populations?
So if you recall, well, the TRPV positive cells were in brown fat. So we didn't. Yeah, so and here we were looking to inguinal, so we didn't see them. We didn't see any expression of 3p1 in the tissue, so I don't think they are present within the inguinal depot.
All right, thank you.